So next up in our workshop is Nick Hasle, who's a graduate student in Doug Fowler's lab here at UW. And he's been working in his graduate work on developing this technology, visual cell sorting, which I'm sure he'll talk about today in this session, which is about extending DMS to visual phenotypes.
Awesome. Thanks, Ian. It's interesting to be up here and actually talking to you guys rather than like muttering into the computer. Yeah, I'm happy to be here today to talk about kind of two methods, one that I developed and one that was developed at the Blaney lab on the East Coast for doing deep mutational scanning on visual cellular phenotypes. And I'm not going to go into too much depth about the actual experiments that were done. I don't really have time to go into that much depth. I'm more going to provide an overview of how these methods work and how they're fundamentally different from each other and what their relative strengths and weaknesses are. So the other thing I'll say is that this is a talk about microscopy, and microscopy is something that like a lot of us don't think about very much. So I'm going to give a little bit of background into that, but if anybody is if anyone's confused or I'm not making sense, just stop me and ask the question there rather than waiting for the end. Okay, so I have spent the bulk of my PhD working on visual cellular phenotypes, and it's become more and more clear to me how awesome these phenotypes are. One thing that's nice about microscopy is you can look at a range of different types of processes. So you can look at protein localization of a single protein of interest. You can look at the morphology of a cell or the behavior of a cell, for example, in migration. And the first of these examples is an example of like studying your protein of interest using kind of microscopy. But the other two I like a lot because they're analogous to a growth-based assay in that they integrate multiple pathways. The multiple pathways can affect, for example, what a cell looks like, or how it migrates. And so you kind of get this integrated readout of cellular function rather than looking at a single pathway, which can be kind of interesting. Another thing about visual cellular phenotypes is there are many clinically relevant genes that impact phenotypes that we can only really assess using a microscope. So I have two examples up here that I think are really interesting. And I think they're interesting because variants in these genes cause more than one disease. So for example, lamin A, a mutation in lamin A can cause cardiomyopathy, it can cause muscular dystrophy, it can cause premature aging, it can cause fat storage disorders, and we really don't understand what the genotype-phenotype link there is. And perhaps by looking at microscopy-related phenotypes, such as this punctate distribution you get of lamin A and some of these variants may help us better understand those links. The second example is up here is PTEN. PTEN can both cause cancer predisposition and autism. And again, the links, the genotype-phenotype map between those two, PTEN and those two diseases is not well understood. And PTEN causes changes in cellular morphology when mutated as well. So I think that Molly and Eric did a really good job of describing how selection methods are used in this sort of a framework. This is kind of how I think about it in terms of doing what we call a pooled microscope-based screen. So you prepare your library like we've learned about today, and then you do lentiviral transduction or some sort of recombination to get that library into your cells. And you have all of those cells in a single dish, which you can then assess using the microscope and assign a score to each variant in your library according to your microscope-related phenotype. So up here I have two cells with different morphologies, and you can kind of assign a score describing how often the variant is associated with that stellate morphology rather than that rounder morphology. Slide. Okay. So there are a few things, pieces of computational expertise you need before you can do this kind of an experiment. One of them is segmentation. You need to kind of know how you're going to actually turn your image into individual objects that can be analyzed. And so here I have some human nuclei, and there are various computational methods that allow you to kind of group pixels into objects, into individual nuclei that you can then study further on. The next thing you need to be able to do is do your phenotypic analysis. This can range from really simple things like fluorescence intensity to really complicated things like morphologies that are assessed using machine learning. And you kind of, if you're thinking about your phenotype that you're interested in, one thing you may want to be aware of is how difficult that phenotype might be to actually assess using microscopy in. And then the third thing that's related to a couple different problems is image or object registration. And by that I mean, in some cases you take multiple images and you need to align cells between the images or spots between the images. And so that's another piece of like computational work that's going to need to be done in an automated fashion. And that's something I guess I want to stress here is that for both of these methods, for these deep mutational scanning methods, you need to be able to do these in an automated way. It can't be human driven because the scale is too large. So I'm going to try to kind of quickly go through these two different methods today. One of them is one I developed in the Fowler lab called visual cell sorting. We posted it on bioRxiv at the end of last year. And the other one is one that's been kind of in the mix for several years now, but was recently put to work in a really awesome way by by David Feldman and Avtar Singh in the Blaney lab. And I'll talk a little bit about what they did as well. So like Molly said, visual cell sorting is a separation-based selection method. And the idea is that if you have a population of cells with two different phenotypes, you can separate the cells into their two respective phenotypes physically from one another. And it's a facts-based separation. And how it works is we express a protein called dendro-2 in each cell in our experiment. And dendro-2 is this photoconvertible fluorescent protein that's green at baseline. And when you illuminate it with UV light, it turns to red. And it does so irreversibly. So if there is a way to illuminate just those round cells, then the dendro-2 in those cells will irreversibly switch from green to red. And that's something that you can see on a flow cytometer and sort for on a flow cytometer. What's really nice about the system is we found that by just decreasing the intensity of illumination or the time of illumination, you can modulate the signal quite well. So if you illuminate the star-shaped cells, for example, for less time, then they also turn red, but not quite so red. And this is what the data look like on a flow cytometer. So I plotted up here the ratio of activated to unactivated dendro-2 signal. And you can see that we can get 4 different bins of activation, i.e. your 4 sort bins, just by illuminating cells for 800 milliseconds on the far right, 200 milliseconds, 50 milliseconds, and then not at all. And so the question you may be wondering is, that's great, but how do you illuminate these cells specifically in high throughput? And the answer to that is we use what's called a digital micromirror device. This is an array of thousands of small mirrors that can all be switched on or off. And so if they're all on, then the whole field of view that the microscope is looking at is illuminated with the UV light. But if you turn some of them off, you get patterned illumination. And the pixel size, each one of these mirrors can illuminate a very, very small portion of your cell. So it can illuminate, these are human nuclei, and it can illuminate inside a single nucleus, which means it's very, very easy to illuminate one of these nuclei without illuminating the other. And so just to put all of this together, what we have is an automated pipeline where we image cells. They're all expressing dendro-2. We have an automated image analysis pipeline that segments those cells and classifies them according to a phenotype of interest. We then selectively activate some of those cells, so we turn their dendro-2 irreversibly from green to red. And then the microscope moves to a new field of view where it repeats that process. And this process is repeated over and over and over again for hundreds of thousands of cells in your dish. And then at the very end, you take all the cells off and you sort cells according to their dendro-2 fluorescence, which now kind of marks your phenotypes. And so in the end, you have cells with one phenotype in one of your sort bins and cells with the other phenotype in the other one of your sort bins. And then to actually analyze the data, you do exactly what you've heard of, what you may have read in other sort-based mutational scans. where you just sequence each of those two sort bins, and then you take a ratio of the variant over the wild type within each sort bin and get scores that way. So it works much like much like other sort-based mutational scans. I did a bad job. That comes later. But this, there are two additional things about visual cell sorting that I kind of want to highlight. So the method that I just described was done in live cells, which means that if you have a dynamic phenotype, your cells may kind of change phenotype over the course of the experiment, which can be problematic in terms of scoring them. However, Sriram and Hyunjin in the Fowler lab have done some amazing work kind of getting this protocol to work in a fixed cell context. So you can fix your cells and then activate and sort them after fixation, meaning that you won't have this problem of phenotypes changing over the time course of the experiment. The other thing that they've done, which is super cool, is they've managed to switch it over to an antibody-based visual cell sorting system, where instead of expressing Dendro2, you have an antibody that essentially does the same thing as Dendro2. And what that means is you don't have to do that in that protein engineering step or cell engineering step beforehand. So just a few key experimental requirements for doing this sort of an assay. The first is that you do need a predefined cellular phenotype of interest. That automated image analysis pipeline that I talked about, that needs to be predetermined by you and probably pre-validated on some pathogenic, known pathogenic and benign variants. The second is that software that you develop, it needs to be pretty quick, because if it's slow, then at each position, at each field of view, you're going to waste time with your computer kind of chugging through it. And that's a problem as well. Maybe it becomes less of a problem if you're using that fixed cell protocol, but still, it's nice to have it as rapid as possible. We found that a lot of machine learning methods are ultra rapid and allow you to do this at very high throughput, which is nice. And the third is you need some commercially available microscope hardware to add to a wide field microscope. And the three pieces are live cell incubation, if you're going to use live cells, a digital micromirror device, and a 4 or 5 nanometer laser, which is the laser that will be used to activate Dendro2. Okay, before I go on, are there any questions about how that works? Well, I have everybody's attention. Yes. Yeah, so we go through about 1 million cells. We can get out about 1 million cells in 12 to 24 hours, depending how fast your image analysis pipeline is. So that's kind of the scale we're working with. Okay, so the other pooled screening method that I talked about today, that I'm going to talk about today, is in situ sequencing. The goal of in situ sequencing is, instead of using a selection method, it's you're basically going to sequence your barcode inside your cells, inside your fixed cells on your slide or in your tissue culture dish. So it's like a fundamentally different way of doing a scan, where there's no selection. You're just phenotyping your cells. You're figuring out what barcode is inside those cells by doing a set of molecular biology reactions, and then you're directly linking your genotype that you just got using your microscope to your phenotype, which you also got using your microscope. And it has 5 steps. It's definitely an involved molecular biology kind of process, because you're essentially going to end up doing like an Illumina sequencing run, but you're doing all the liquid handling yourself, and you're doing all the imaging yourself, and you've got your own image analysis that you're doing. So you start out by fixing and permeabilizing your cells. You then do a reverse transcriptase reaction, and that reverse transcriptase reaction will capture the barcode that is associated with your gene. So in this case, the barcode is in orange and the gene is in black. I think that, yeah, we'll see that there. So you have your gene of interest on the left, you have maybe a 12-mer barcode, and then you have that's barcode is kind of inside your 3' UTR. And so you do a reverse transcriptase reaction to kind of turn that into some cDNA. You then use what's called the molecular inversion probe to capture the barcode sequence. So this is a probe that binds both downstream and upstream of the barcode. And then you can use a polymerase to fill in and capture the barcode inside that molecular inversion probe, and then a ligase to ligate it shut. And the reason you do this is because it allows you to amplify that barcode using a subsequent rolling circle amplification reaction. So this is where a primer binds to now your molecular inversion probe that's captured your barcode in a highly processive polymerase, and it goes through and makes many, many, many copies of that barcode. This is critical because you're going to be reading out this barcode using the microscope, and that barcode thereby needs to be amplified so you get enough fluorescent signal per base. What you do next is really incredible, I think, and that is where you add fluorescently labeled nucleotides that you get from a MiSeq kit, and do the same sorts of reactions that a MiSeq kit does during a sequencing run. So what this looks like is shown up here, where on the right we have some nuclei that are stained, and we add you kind of add in these nucleotides and then ligate them onto the barcode, and then wash away the excess nucleotides and image. And each one of these spots is one of those rolling circle amplification reactions that worked. And then you add your nucleotide, you wash away the remaining ones, and then you image, and you can see, for example, in cell 1 up there, the barcode reads a C, and in cell 2 it reads a G. You then strip off the fluorescence and repeat this step over and over again to kind of sequentially read out your barcode. And then the analysis is a little bit tricky, but basically you have your segmentation pipeline that gives you a list of cells. You have your variant that you've read out using your barcode. And then you have some sort of cellular phenotype that you've also measured and scored. And what they did in their paper is they essentially took some controls, which in our case would be like a synonymous distribution of variants. And then they had their quantitative phenotype, which in this case was nuclear translocation. And they then looked for variants that were significantly outside of that distribution of controls and scored those using like kind of how far outside of that distribution they were. Okay, so for this, for in situ sequencing, one of the things to keep in mind is that each run requires some pretty expensive molecular biology reagents. You need reverse transcriptase, you need a Phi-29 polymerase, you need some MiSeq fluorescent DNTPs, so it's a little bit kind of more expensive reagent-wise. You do need a barcoded variant library because you can read 12 nucleotides and not really any longer than that. So if you're hoping to read out a tile of a library, for example, it's not going to get you there. And the final thing I have up here is good hands. I just put that up there because there are a lot of steps. There are a lot of washing steps. There are a lot of reagent addition steps. And because the reagents are so expensive, you really don't want to mess up too many steps or you're going to end up costing yourself more and more money. So those are kind of the things to keep in mind there. Okay, let's see. Okay, so I thought I would end with kind of what I think is the kind of higher level pros and cons of each of these two approaches. One important difference is, like I said before, visual cell sorting, kind of you need a predefined phenotype that you're interested in going after. So that you kind of have your analysis pipeline that runs during the experiment that identifies that phenotype. In situ sequencing is nice because you have the images connected to your barcodes. And so you can go back and do other analyses on those images to connect like new phenotypes. Visual cell sorting takes less time per replicate. In situ sequencing, you get more cells per replicate, particularly if you can pack them really tightly together in your assay. And so they kind of are a wash in terms of like cells per day kind of a thing. Visual cell sorting is less expensive per replicate because you can kind of pool all your replicates in a single next-seq run rather than having to use those expensive reagents per run. But in situ sequencing has kind of fewer hardware requirements. You can do those in situ sequencing reactions on a wide field microscope. One of the two things that really put these two methods apart from each other, I would argue, is that visual cell sorting can actually be used with any downstream assay. So if you have to sequence more than a 12-mer barcode, then you can do that with the visual cell sorting. If you're interested in the phenotype for other reasons, like Perhaps if there's a variant with incomplete penetrance that you're interested in understanding what's going on there. You can study that with visual cell sorting because you can subject cells that have the phenotype from cells that don't have the phenotype to proteomics or transcriptomics. And it's pretty flexible in that way. In situ sequencing, you know, it gets that 12-mer barcode, it gets your barcode, and so it's more limited in that way, but it's a single cell assay. And that means that like per replicate, you're checking the phenotype of multiple cells with the same barcode. And you can kind of get an idea of what the distribution of effects of that perturbation has per replicate, which you don't get with selection-based methods. With selection-based methods, we always get one readout for each variant per replicate. And so I think that's a pretty key advantage to think about as well. Okay, I guess I just want to make some acknowledgements to Doug and Sriram and Hyunjin, who've done a lot of the work that I talked about pushing visual cell sorting forward. Tony Cook, who is the rep at Leica we worked with to get this system up and running. It was like we were not a microscopy lab at all when we started, and we managed to get it working pretty quickly. And then Emily and Heather for a collaboration we've done with them, and Avtar, who is one of the co-first authors on the in situ sequencing paper, and who's been really helpful in terms of thinking about whether we should set it up in our lab. And with that, I'll take any questions.